Okay, good afternoon, one and all present over here. Welcome to the second session for the day. We have today Dr. Rahul Parikh, who is Assistant Professor of School of Science, Dabasha University. Okay, he will be dealing with the topic stress physiology. Okay, and he has done his PhD in applied ecology, and he has also passed CSIR-NET exam. So he will be able to tell you what questions that you can expect in the NET exam. Okay, over to you, Rahul.
Thank you, ma'am. Good morning, students. I'm Dr. Rahul Parikh, as ma'am said, Assistant Professor at National University. Today, I'll be speaking about stress physiology that mostly concerns with the plants. So, basically, not this stress. We'll be discussing this stress, that is a plant stress. If you define plant stress, it is basically refers to an external condition that adversely affects the growth and development of productivity of the plants. It triggers a wide range of responses, like it can alter gene expression, it can you can say alter cell metabolism, it can change in growth rates, it can change in you can say crop yields, and many similar aspects. If the stress level is mild, the plant can tolerate. If the stress increases, the death of the plant. This stress can be due to either abiotic factors, so that's temperature, it can be a water, it can be salt, either if in mild, if this stress are, they can be due to one source, so it can be due to either temperature, or it can be due to water stress, deficit of water or flood of water, cold stress, cold climatic stress, hot climatic stress, salt stress, or in very simple terms, for a few of the places, I can also represent as sodicity stress rather than salt stress. So either of this can occur individually in mild condition, but when they start to gather up, they causes a severe effect. So that is abiotic factors. Biotic factors, it can either cause by pathogens or it can cause by pests. This pest can be any insect or nematode or any gas. For pathogen, it can be a fungi, it can be a bacteria, it can be similar materials, even viruses. Okay. So whenever we are representing stress, an acute response come to its place. So either the stress is abiotic in nature or biotic in nature, it will cause some response in plants. This response can be categorized into three aspects. Either the plant is resistant to the stress, the plant is susceptible, or the plant is able to avoid the stress. Kind of, this is an evaluation which in raise after certain modification in plants. So if you take resistance, there are many plants which over a period of time acclimatize to the situation or to the responsible stress. Once they are acclimatized, they continue their growth and they can survive. For example, I can give you is you can represent all the plants which are living in a desert condition. A very effective example. They all have acclimatized to the mild water stress as well as heat stress. So they have certain modifications. They have this ability to adhere or to sustain or to resiliate this stress. So they are able to acclimatize, they are growing very successfully, and they are surviving. The second is susceptibility. Susceptibility is caused in plants which are weaker in nature. They can't or they don't have capability to resiliate to stress. Such plants undergo senescence, and post senescence they die. Some of the plants or susceptibility also depends on the quantum or intensity of the stress. If there is a mild heat, the plant can sustain. But if the heat level increases, the plant may not able to sustain. Similarly, if you can say drought is there, then plant can sustain drought up to few days or months. But if the drought condition increases, the plant may not able to sustain. So depending on intensity, the susceptibility of the plant changes for different stress, and on basis of this susceptibility, the plant undergoes senescence or death. And the last comes avoidance. A very common example I can give is ephemerals. They come in particular season where the conditions are appropriate to them. They grow in this condition, they avoid all kind of stress, and they survive. Okay. So uh, out of different stress that can be incurred due to, you can say, abiotic factors and biotic factors, first I will be talking today about abiotic factors. So if you consider abiotic factors or abiotic stress, is caused either by, as I represented, temperature, that can be heat stress or cold stress, water, so drought or flood, and last is salt. Heat stress and light stress or radiation is, or you can say irradiance rather, not radiation. Heat stress and irradiance is working hand in hand. So first, just depicting the abiotic stress. Uh, I hope many of you will agree with me that this is a diagram which is representing basically CO2 concentration to irradiance. Okay? As we see, with the increase in light, with the increase in CO2, the plant may increase its photosynthetic ability from to certain, then it gets saturated. Over a few period, it remains saturated. At this place, the plant is undergoing stress. This stress is not as high that it causes any necessity or important morphological disturbance which can affect the plant. So at till this place, the plant is tolerant in nature. But if the irradiance still increases, then concentration is increasing, may not able to photosynthesize. Rather, it will go to exactly invert condition, that is photoinhibition. It will stop the photosynthesis even the CO2 is present. This condition may vary for C3, C4 and CAM plants. As we know, CAM plants are partially, you can say, adapted in nature. The limit of saturation may increase as compared to C4 as compared to C3 plants. So if I go in the decreasing order of, you can say, tolerance level, then the first comes is CAM plants, the second comes is, you can say, C4 plants, and third comes is C3 plants. Okay, rather than going for this, okay. So basically, as explaining, as I said, tolerance. So if we take CAM plants, they are basically tolerant in response to the many morphological characters they have adapted. For example, thick cuticle, one important character which, you can say, leads to reflectance of most of the light or irradiance from the leaf surface. In addition to this, they have capability to throw back the light in a form of infrared radiation. So this is one of the, you can say, adaptation. In addition to that, CAM plants basically absorb atmospheric CO2 at night, then it closes its stomata in the day period, and then it performs a photosynthesis in daytime. So in a way, they are kind of regulated. They are capable, or they are already prepared for some of the irradiance, you can say stress or light stress. Then comes C4 plants. So C4 plants, if you take, they have a specialized mechanism of absorbing CO2. So they fix CO2, then they provide it to the photosynthetic cycle or C3 cycle. So in a way, they are a bit more, you can say, capable, or they are a bit adapted to the heat stress. Kind of, they are partially tropical plants. You can represent them. Last comes C3 plants. C3 plants are very sensitive in nature, and if you consider photosystem, PS2 is very sensitive in nature to the irradiance. As the irradiance increases, the damage, first line of damage is caused to the photosystem 2. So once the irradiance increases, the PS2, or rather C3 plants, stop photosynthesis, and it starts a process which we refer as photorespiration. So for few week time period, they continue photorespiration. This is a adaptation to tolerance. Once the irradiation increases before beyond its you can say capability, then the plants undergoes photoinhibition. This photoinhibition is basically by de-integration of PS2. Sorry. Okay. So if you take a basic stress, like stress, then as I showing you in the slide, as I said, PS2 the irradiance. So active PS2 absorbs, you can say, photosynthetically active radiation, or PAR. Yeah? It absorbs certain limit of PAR, but if this irradiance level increases or intensity increases, then becomes inactive. This process is reversible. So you can say if the irradiance increases, the PS2 system will become inactive, and it will stop for you can say photosynthesis. But the damage will not be that much that it can't be reversed. They will be, or they possibly can be, reverse the process, and again the PS2 system will get functional. But if in case this damage increases beyond limit, then it will cause a damage in you can say PS2 system. Once the damage has incurred, the PS2 will disassemble. It will basically disintegrate, or will separate out from the PS2. One specific protein which is responsible for this, or which is damaged in this whole process, is D1 protein. This D1 protein gets phosphorylated, and once it's phosphorylated, it is marked for degradation. So the D1 protein is degraded, and it is removed from the segment. With the degradation of D1 protein, the gene is, you can say, activated, and new one D1 protein is synthesized. It is inserted in thylakoid. The PS2 system gets reassembled, and the active photosystem is prepared, or is active, rather PS2 system is activated, and once again the system continues. This I represented for photosynthetically active radiation. The same thing happens in UV radiation. So if UVB or UVA increases in number, or its radiation increase, then the same thing will happen. The D1 protein in the PS2 will get degraded, and it will be removed. A new PS system will be produced, and it will be entered into the system. So this is again the same thing. The DA1 repair cycle, specifically for even repair cycle. So functional PS2 excess light, it goes to reaction center damage. PS2 disable self or disassembly happens. D1 disintegrates. A new D1 is synthesized, and as I wrote in blue, as written in blue colors, the gene which is responsible for synthesizing the D1 protein is PSBA. This gene will be activated and it will produce a new D1 protein which will be inserted into PS2 system, will get rearranged and it will become functional. Okay. The question came out. Okay. The question comes, if a mild heat stress is incurred, will the plant be able to tolerate it? Yes, there are many, you can say, possibly there are basically two set of lines or two line of defense which protects the degradation of D1 protein, or which protects the degradation of PS2 system. So this is the first cycle, which is you can say violoxanthine cycle or xanthophil cycle. Yeah. Okay. Before before continuing further, just one question, just one explanation. When we take proper system 2, yeah, or rather we take the whole system, there will be a section which is referred as antenna, and the second part is referred as reaction core. This reaction core is having all the or rather we take the whole system, as we energy is absorbed, or rather the excess energy is
Ma'am, can you please tell
points.
sir that he
This
is
is
not
represented
audible
the two
in the
examples at each
video?
other, as represented depending on the virus and thing with this part and parcel of you can say quenching mechanism. So whatever the additional photon is coming is quenched in this way, and they are interconverted into different products. So by this process, the excess photon is increases the pH of cell, or rather to the very specific thylakoids. With an increase in thylakoids, and with this increase in stress, a metabolite known as ascorbate gets. Sorry. Okay, my voice is not. Uh, am I clear now? Am I clear now? Uh, am I okay? Great. Okay. So as I said, with the advent of heat stress or light stress, rather high intensity of light, the pH of the plants, yeah, the pH of the thylakoids decreases. This decrease in pH is led by ascorbate, or rather, in very simple terms, this pH decrease in pH is induced by induction of ROS, reactive oxygen species molecule, which leads to increase in ascorbate, that is, antioxidants treat the ROS species. So, with the decrease in pH, the thylakoid cycle initiates, that is, violoxanthin comes to astaxanthin and to zeaxanthin. Once this stress is removed, the pH again regains normal to 7.5, and the normal process which happens, or the zeaxanthin back to violoxanthin. Okay. So as I said, there are two defense mechanisms before the whole system becomes inactivated, or the PS2 system completely becomes destroyed or damaged. So as the picture is representing, the system gets excited and de-excited as the basic model of, you can say, process of photosynthesis. But if the process or the heat radiance or light irradiance increases, then the first step of defense comes into place, that is photoprotective energy dissipation. This photoprotective energy dissipation occurs by two processes. First is light harvesting. So as I depicted earlier, basically in, you can say, this complex, the first the photon is absorbed by, you can say, antenna molecules, which has high concentration of carotenoids and xanthophylls. In very simple terms, you can represent them as sun protecting film for plants. Clear? So they absorbs the photon. This photon absorbed is, you can say, converted or is quenched by zeaxanthin cycle, or you can say this energy is released in blue light, is converted to blue light and is released back. Right? So kind of fluorescence happens, or zeaxanthin dependent quenching happens, or state transition happens. So in short, the light is harvested in, or then is quenched, or else it is, you can say, dissipated by electron transfer. So leads to cyclic electron transfer, which is a basic method. It transfers the electron to the further lineage and it gets to the photosystem 1, or it transfers the extra energy to the alternate electron. Most commonly, this alternate electron sink is oxygen. So either it, as I said, it transfer quench harvesting commences, or it quench, or it transfers the electron to the alternate electron sinks. So that is first line of defense. If that fails and additional energy is generated, then this additional energy is taken up by again oxygen, but high, you can say, reaction, or in simple terms, it initiates reactive oxygens. It synthesizes many reactive oxygen species. Once this additional reactive oxygen species are generated, the photosystem or the chloroplast will induce gene regulation and will produce ROS scavengers and antioxidants. So as I represented earlier, ascorbate is generated. So uh, related to the ascorbate, Milius enzymes are released. They will quench the reactive oxygens present in thylakoids. If and only if this defense system is failed, then the excess irradiation will lead to damage of B1 protein and thus will lead to photoinactivation. So the barrier which I represented to you, that there is a few space, or there is a few area, there is a part area where even if the irradiation increases, the plant will partially continue to perform photosynthesis with no effect of increasing CO2. So at that point of time, this defense response comes into place. So up to certain light intense light intensity increases, the plant can quench it either in a form of fluorescence or in a form of CSI, in a form of excess or alternative electron sinks. But if the intensity goes beyond this, then the plant will you can say, inactivated, and the B1 protein, which is a very important part of the photosystem 2 denatured, will get degenerated. Post stress, a new D1 will be produced and will be connected back. But by the time it gets photo inactivated, and once the photo inactivation goes beyond its reversal, a chronic inhibition happens. The net photosynthetic capability of plant is lost. So that is mostly, you can say, the effect of light stress. To this, there is many acclimatization in plants, or you can say there's a tolerance response which induces in plants. There are two light dependent hormones, or rather proteins, which I'll represent: phototropins for light irradiance tolerance in plant, phototropin 1 and phototropin 2. So if the light intensity is higher, you can say, then both phototropin 1 and phototropin 2 will come into place, and they will mobilize the chloroplast in cell. Clear? And if you can say the light intensity is low, the phototropin 2 will rearrange the chloroplast in normal manner. So if you see in the slide, as you can see in accumulation response or weak light, rather the chloroplast are arranged on the entire lateral, or you can say dot surface of the cell. But with the increase in light irradiance, or with the strong light when it falls onto the cell, it goes onto the side of the, or it goes to the corner of the cell. Thus, it avoids the additional photon from being absorbed in the cell. This is a light dependent movement which happens in the cell in response to phototropins. So, stress effect, light stress inactivation, the D1 protein gets degraded. Once the stress is over, or the stress is removed, again the new D1 proteins will be synthesized, and with the new D1 protein synthesized, the PS2 system will again become functional. So the cycle will continue. To adapt this, the plant have synthesized a movement of the whole chloroplast in response to the light. So as you can say, accumulation response and avoidance response in response to the irradiation caused by, or the movement initiated by, phototropin. So this was the first stress, that is light stress. The second stress comes is temperature stress, or in temperature stress, high temperature, that is heat stress. Heat stress comes hand in hand with the light irradiance. Higher the light irradiance, higher the heat will be, and thus it will, or you can represent them as a combined effect. Thus, you can say, so basically heat stress caused in plants by high temperature. Again, this heat stress depends on the biome in which the plant is growing. If the biome is, you can say, or the plant are adapted in temperate places, the heat stress may start from the very low temperature, that is around, you can say, 30 degrees Celsius or even lower than 30 degrees Celsius. But if the plant is growing in tropical area, then this stress trigger may start from around, you can say, 40 degrees Celsius or 45 degrees Celsius. In case of tropical plants, the basic temperature tolerance degree is 45 degrees Celsius. Hydrated tissues or living matters are more prone to heat stress. Non-parts like seed pollens can tolerate heat. Dry seeds, they can go up to 120 degrees Celsius and they can remain active, or they can, you can say, viable. They remain viable. Same is with pollen grains. Pollen grains can survive or endure up to 70 degrees Celsius. The temperature beyond that may, temperature beyond that may inactivate them. I'm again repeating the word may acclimate at high temperature. They are all known as thermoregulant or thermotolerant plants. Okay, so if you I categorize them, basically, the plants can be a saprophytes, the plants can be a mesophytes, the plant can be a thermophiles. Saprophytes are basically the plants which are adapted to survive in heat tolerant species. Okay. So if I take the heat stress, if you go for a normal physiological impact which happens on plant, it varies from inhibition of seed generation to reduction of plant growth to improper development of the whole plant, alteration in photosynthesis, alteration in phenology. Alteration in phenology means the leaves shape may change, the number of leaves may change, the, you can say, flower, you can say, the size of the leaf may change. So does with the flowers or inflorescence. Even the season sometimes, or the flowers, the plant does not go into inflorescence condition if the heat stress is there. Alteration in tripartitioning. This is again one of the most important aspects. The next, water loss, yield reduction, reduction of crop quality, and last is oxidative stress. Reduction in crop quality, in a sense, it represents with the advent of heat stress, the plant stops photosynthesis and it starts photorespiration as well as reproduction. So all the produced carbohydrate present in water in the plant is utilized, which reduces the sweetness of fruits. So the quality in a way changes. These are the basic effects that caused by heat stress. If I okay, if I go at cellular level, the first thing that will happen is it, the altered physiology will heat stress will lead to increase in membrane fluidity. The increased membrane fluidity can cause ion leakage. This is due to the disruption of hydrogen bonds and increase in electrostatic interaction. So alteration of electrostatic interaction and decrease hydrogen bonds fluidity, and thus causes ion leakages. So Much background disturbances. Uh, Give me a minute. Okay, so the membrane fluidity the increase in membrane fluidity, as I said, ion leakage increases, and disruption of basic all the membrane based processes. So starting from photosynthesis to respiration to all the processes which are governed by membrane, they all get disturbed. So that was the first effect. The second effect was, or the second effect it causes is the protein loses its three structures. So it may get misfolded or it gets solubilized. So causing precipitation of that protein. This can become lethal for cell. If proteins become soluble, the cell may undergo death. So most important impact that happens to misfolding is rather can go for all the examples. One of the very specific example is PS2, the protein which I reported a few minutes back, that is E1 protein or PS2 core. The proteins in PS2 core gets precipitated due to heat stress, and thus system is inactivated. Next aspect comes is synthesis of RO reactive oxygen species. Okay. So that was effect it caused. The next aspect comes is acclimatization to high temperature. The first aspect is closure of stomata. The second aspect comes is large xylem vessels. Third aspect comes basic cuticles. And the last comes leaf shape alteration. Okay. To continue further, can anyone give me an example which is caused stress, or rather what uh, can become in leaf shape alteration? Any clue? Specifically to monocots. What happens to monocot leaves in presence of high irradiance or heat? Any clue? Am I audible? Anyone with the answer? What happens to the leaf surface of monocots in presence of heat stress, or you can say high light irradiance?
Sir, they kind of shrink and try and turn yellowish color, I guess. Like, they shrink, they cannot retain they lack the water retaining capacity due to heat stress.
Shrinking comes after the leaf falls and a new leaf is developed. So new leaf will be possibly shrink. But
Yes.
what about that leaf? What can happen to that leaf? Okay, so there's a adaptation which is present in monocots. I hope many of you will agree with me. They are referred as bulliform cells or mortar cells. These cells are responsible
Yes,
for
sir.
curling of leaf. So that is one of the way wherein it can reduce the surface area, thus to reduce the heat absorbed or the light irradiance. The next aspect, the first, as I represented, closure of stomata. We all know a very good example, that is granulin acid metabolization plants, called the chem plants. They close the stomata during daytime and open the stomata during nighttime. So whatever the gas exchange happens at the night time. So that is one of the very acute, you can say, adaptation or acclimatization which has incurred in cambrans. The second comes large xylem vessels. This adaptation is possible when the heat stress but is present near a water body which is filled with water. So if the plant has ample quantity of water, then the size of xylem vessel can be increased, a higher quantum of water can be loaded, and through transpiration the heat stress can be reduced. The next step comes is vaxicuticles. Vaxicuticles reflect back all majority of the radiance, or you can say light, which falls on the leaves, so as to decrease the heat which increases in the leaf. The higher the stress or heat, the thick cuticle leaf will have. And the last, as I said, leaf shape alteration. Okay, so that was acclimatization. The next aspect comes is tolerance mechanism. When a plant is undergoing heat stress, it is very common that the plant is also facing water stress. If water stress, the plant may also face salt stress. Clear? So we can represent them as a domino effect. With the increase in heat, the transpiration increases. With the increase in transpiration, the water stress increases. With the advent of water stress and with the advent of higher transpiration rate, the salt stress increases in the plant. So the first three aspects, that is iron transporters, Lea proteins, and osmoprotectants. This you can regard with any place or any aspect where desiccation like symptoms come into play. May it be a heat stress, may it be a cold stress, may it be a water stress, or may it be a salinity stress. The first three tolerance mechanism will be, you can say, present in specifically all type of stress, because they are in a way interrelated. So ion transporters, this helps in stabilizing the membrane proteins and thus stabilizing the ion leakage. The second, Lea proteins, they are desiccation responsive elements and they helps in decreasing the effect of heat stress. The next comes osmoprotectants. Osmoprotectants try to keep as much as water in the cell so as to reduce a desiccation type effect caused by the heat stress. Okay. Um, Basically, can anyone tell me which is uh, why cactus or granulin plants in terms of water are able to survive in desert? What it do to survive in desert? तुम में से कोई पानी डालने जाते हो? I hope not. So what the
So
plant does?
the
What kind of
adaptation is
adaptation?
done in such a way, the adaptation is done in a such a way that firstly, the leaves can be converted to thorns. Then they adapt themselves
Okay.
in such a way कि पानी water retaining capacity can be increased. Like loss of water would not be so frequent compared to the normal plants.
Rather, do they store water?
No, not water. They store certain like chemicals or certain compounds which can help them to survive even in excessive heat and excessive water loss conditions.
Basically, they convert all the available water into acids and alcohols. So they don't have the water to get means to loss. उनके पास पानी होता ही नहीं कि वो water loss कर सके. Whatever the water is converted into different forms. One of the very example you all can accept is if you cut aloe vera, you will never get water. You will get a gel. If you dry that gel, it will become a thin papery. If you add water, it will get hydrated. Clear? It doesn't lose water. Clear? So, in a way, this kind of plants have adapted its whole metabolism in a way that they have converted or they have produced many osmoregulants. The most common osmoregulant I can represent in case of this is you can say sucrose. One, in case of heat stress, most commonly comes is sugar alcohols. So all the sugar alcohols are present in a basic liquid form in plants in normal condition. But if once the heat stress increases, this gets dried up, becomes a wet, thin, papery structure present in water. They don't undergo any morphological change that can cause damage to the plants. Yeah, so these are the in addition to this, they increases the solid potential so as to increase the water potential. This increased water potential leads the cell to absorb as much as water possible into the cell. Once it is done, it stores all the water into the vacuole. The next aspect comes is antioxidant defense. So a very basic thing, with the increase in temperature, all the plants will undergo a process which will or it will undertake a stress mechanism which will increases the reactive oxygen species in the plants. To defend from reactive oxygen species, plant produces antioxidants in the system or in the cells which help to incur or which help to defend against this ROS. Okay, so आयन सपोर्टर्स बन गए हैं, लिया प्रोटीन्स बन गए, ऑस्मोप्रोटेक्टेंट्स रिलीज हो गए, एंटीऑक्सीडेंट्स रिलीज हो गए. The next most important aspect comes is signaling, you can say genetic aspect, so that factors involved in signaling cascades and transcriptional controls which are essential, significantly essential to counteract the stress effects. And the last will come is heat shock proteins. So, so as I represented, there will be many antioxidants which are present. These antioxidants may be an enzymes or they may be a metabolite. For enzymes, example is superoxidase dismutase, which play important role in cellular defense mechanism against loss. Glutathione reductase, which is an important enzyme for ascorbate glutathione system. It maintains a balance between reduced glutathione and the ascorbate pool. Basically, helps to manage hydrogen peroxide. Ascorbate peroxidase, it helps to detoxify hydrogen peroxides in the ascorbate glutathione pathway. It utilizes ascorbic acid to reduce the peroxide to water and monodehydroascorbate. This monodehydroascorbate is again a part and parcel in or responsible in signaling mechanism. The next comes catalase. As represented, it decomposes hydrogen peroxide to the water. The next part comes metabolites. The most important metabolite, or few of the most important metabolites are here. That is, first is alpha tocopherol. It basically scavenges the free radicals in combination with other antioxidants. It protects the structure and function of PS2 as it is as it chemically reacts with the oxygen in chloroplast. So this chemical specifically is synthesized and is transported to chloroplast in plants to help it incur the heat stress in chloroplast. It helps in membrane stabilization and elevates the tolerance of plants during oxidative stress. Okay? The next comes ascorbic acid as represented earlier. In the presence of enzyme, ascorbic acid is used to quench or it's used to destroy the hydrogen peroxide. The same does glutathione. Carotene. So rather carotenoid. Carotenoid runs in the same manner as I depicted for the light stress. So all the xanthophylls and carotenoids which are present in the plant or which are present in the chloroplast, they act as a sunscreen lotion for the cells. They absorb the additional irradiance or heat from the cells. They convert it in. They convert to different products. Thus, you can say in a way it quenches the heat and the light. So, in a way, they are first line of tolerance in heat stress. Okay. The next step comes is genetic. So signaling cascades and transcriptional control. With the advent of high temperature, oxidative stress increases. This oxidative stress leads to disruption of membrane properties, protein enzymes, and cellular homeostasis. Once the membrane protein is disrupted or once the membrane protein is disturbed, they will induce transcriptional factors. Mostly these transcriptional factors are calcium dependent protein kinases or mitogen activated protein kinases. So CDPKs and MAPKs or MPKs. MPKs can also be represent. You can say you can add number of Ks. So MPKs, MPKKs, MPKKKs. They all are you can say comes in sequence to each other. Okay, second. Okay. So that there leads to transcription, and these are transcriptional factors which activate stress responsive genes. This stress responsive genes, specifically CDPKs, and rather the CDPKs and MPKs or MAPKs, they are responsible for you can say enhancing the synthesis of antioxidants, either enzyme or metabolites or both. Once the antioxidant enzymes are activated, free radical scavenging is done. Signaling molecules and osmoprotectants are also released to you can say manage the or balance the effect of heat stress. This arose toxification, reactivation of proteins and enzymes, re-establishment of cellular homeostasis, and thus the heat tolerance is developed in plants. Okay. The last most important aspect is HSPs. Clear? They are referred as heat shock proteins. Clear? One of the most important aspect of heat stress in case of plants. In plants, basically HSPs can be divided or grouped into 5 different families. The first one is HSP 100, or the gene is CLPB. Second is heat shock protein 90, heat shock protein 70. Heat shock protein 70 can also be represented as DNAK, that is a gene name. HSP60 or GROE. The last is HSP20, or they are also represented as small heat shock proteins, or SHPs. Okay, the first four, rather, there will be one more which will be coming. So HSP 100, 90, 70, and 60. They are very specific to heat stress. HSP 20 or small heat shock proteins. They are not very specific to heat stress. They can also be induced under condition of either oxidative stress, drought stress, flood stress, cold stress, light stress. Clear? So they are kind of very common in nature. They can be induced in different systems. Okay. Heat, the heat shock proteins and their function. So HSP 100, they are ATP they function. Their function is ATP dependent dissociation and disintegration of aggregate proteins. So all the proteins which are you can say unsolubilized or insolubilized, or gets precipitated or gets misfolded. Yeah? This type of ATPs are dissociated and disintegrated by HSP 100, HSP 90. They are co-regulators of heat stress linked signal transduction complexes and manages protein folding. Again, they require ATP for the function. Few of the paper depicts that HSP90 is not, or has not been very specifically derived from the plants. So its activity is not specifically explained in the plants. So one school thinks they are not specific to plants, one school thinks they are specific to plants. Okay. The next comes HSP70. HSP70 and HSP60, they are two heat shock proteins which are very reserved or conserved in nature. Mostly HSP70 will have same structure around the organism. So if I take different organism, will rather have the same type of structure in a sense. Okay, so HSP70 and HSP40, they are they function as primary stabilization of newly formed proteins. Again, ATP dependent binding and releases. The next comes HSP60 and HSP10. Same ATP dependent specialized folding machinery. Of this, HSP60 specifically is, you can say, in relation to Rubisco. 60 is responsible for properly folding of Rubisco molecule or Rubisco enzyme for its functioning. One of the very specific effect which is caused by heat stress is that the Rubisco binding enzyme, smaller subunit and larger subunit, they are not properly folded, and thus the Rubisco can't become functional, thus remains inactivated. This enzyme, rather this heat shock protein HSP60, is responsible for properly you can say folding of Rubisco enzyme and thus functioning of photosystem. Okay, the last comes HSP20. It is responsible for formation of high molecule uh, weight oligomeric complexes, which serves as cellular matrix for stabilization of unfolded proteins. HSP 100, HSP 70, and HSP 40 are needed for its release. So this was all about heat stress. The next aspect comes is cold stress. Okay. Okay. So as we discussed for heat stress, the same comes is Cold stress is exactly the opposite. It is caused by low temperature. Again, depending on which biome, the effect changes. If the plant is in temperate conditions, they have a more, you can say, tolerance towards cold. If the plant is growing under tropic condition, then they have less tolerance towards cold. The same as you can represent for the heat stress. Hydrated tissues or living matter are more prone to cold stress. If the cell is having water and is induced for cold stress, either, excuse me, they can go for chilling, or even bad, they can go for freezing. Yeah? So they are very prone, hydrated tissues. Non-hydrated parts are partly tolerant. Some of the seeds can be grown below, or they can endure temperature below 0 degrees Celsius. Some of the species can accept, you can say, cold temperature of absolute zero. So that is minus 273 degrees Celsius with no impact. Right? So again, hydrated tissues are, you can say, prone to cold stress. Non-hydrated parts are not prone to cold stress. Depending on temperature conditions, either the plant may undergo chilling injury, or it may go freezing injury. Chilling injury in a way is caused for a temperature range between 10 to 15 degrees Celsius. So if the plant is tropical in nature and the temperature goes below, you can say, 15 degrees Celsius, it causes chilling injury. Um, in a very specific terms, it can go for, you can say, ice freezing of leaves, inactivation in photosynthesis, inactivation of respiration, and many such processes. Basically, the enzymes will stop functioning. Same as in case of heat stress, the same will remain in cold stress. But if it goes below 0 degrees Celsius, then it can go for ice formation. Once the ice is formed in cells, the plant will die. What effects does it cause in the plants? The first is a primary effect you can represent is membrane destabilization. The second is water potential reduction. The third is cellular dehydration. The fourth is ice crystal formation in cells. Secondary effects it can cause is reduced cell or leaf expansion, reduced cell and metabolic activities, stomatal closure, photosynthetic inhibition, leaf abscission, membrane and protein destabilization, iron cytotoxicity, cell death, physical destruction, and oxidative stress. Okay. Before going further, one thing I like to ask to you: heat stress. It is understandable that it causes desiccation, but how cold stress causes desiccation? Can anyone answer this? Why cold stress causes desiccation in plants? Any clue? Am I there? No. Chalo. Am I audible, by the way? Okay. Am I audible? Anyhow, no response. Wow. Okay, going further. So, as I represented, you can say, as I represented, in case of heat stress, the membrane becomes fluid in nature. In a sense, the you can say from a gel conformation, it becomes a very liquid conformation. In case of cold stress, from gel conformation, it goes for a solid conformation. In a sense, it gets solidified. So rather than you can say, as you can see in heat stress, due to very decreased bonding or decreased hydrogen bonding and increasing electrostatic repulsion, the membrane starts separating. Exactly inverse happens in case of cold stress. The cell wall starts to get shrink in nature and gets solidified in nature. So in a way, the cells start shrinking. Okay? This again causes many issues, same as it causes in heat stress. वहाँ पे गर्मी के कारण वो we can say function नहीं कर पा रहा था, यहाँ पे ठंडी के कारण वो function नहीं कर पाएगा. There you ion leach happens due to disruption of wall integrity. In this case, the ions won't be able to, you can say, circulate it, or it won't be able to pass through cell membranes due to inability of plasma membrane to function. The proteins will get solidified, the fluid nature of proteins will stop. Once this, you can say, function is stopped, the membrane interaction will stop. So incoming and outgoing of ions is hampered. Incoming and outgoing of different molecules is hampered. So in a sense, the functionality is hampered. The same as I said, cold stress or heat stress will result in almost same type of effect, clear? Molecular effect. Okay. Now what happens due to cold stress? Freezing. If you take an outer cellular structure, clear, on plants, there will be many ice crystal formation on leaf surface. There are many nucleators. Nucleators are basically, you can represent them, the causal effect in a way. So this nucleators, example in case of ice crystal formation of leaf surface, the nucleators are few of the bacteria which remains or which grows on the leaf surface. They remain on the leaf surface. They are a kind of ectoflora which are present on the leaf surface. Example is Pseudomonas syringae or Irvinia ricola. Clear? First one is bacteria. The second one you can represent as fungi. They causes nucleation, leads to ice crystal formation on leaf surface. So I hope many of you have seen this as a I can say screen shaver, or in many of the images there will be a ice which will be formed on the plants. Right? Most common aspect in the temperate conditions or in the cold days, the whole plant is covered by ice. This ice crystal formation is induced by nucleators, and that enhances the effect. So this freezing is caused on the plants. Till part it is not toxic in nature, but it has no impact or at most impact on the plants. It will cause some damage, but that damage is, you can say, manageable. It can be revert back. The next part comes in plant freezing. This can be extracellular, this can be intracellular. For extracellular, one very thing, the apoptic water which is present freezes. Yeah? This again is not that much harmful, or rather it's harmful but that not dangerous. the system can be reverted back, and the in, you can say whole aspect can be managed. The next aspect comes is intracellular freezing or intracellular crystal formation. If this starts in plants, the plants will surely die. They won't sustain. Clear? One of the very technique of cryopreservation. Clear? We completely freeze the plants. But in both of this, we use some techniques to decrease the formation of ice crystal. Clear? Okay. In case of you can say this ice freeze or ice crystal formation within plants, the nucleators are proteins. So many large proteins, they act as nucleators and they start freezing. The whole physiological process is known as nucleation. Now, nucleation is a process wherein almost thousand or more than thousand molecules of water start crystallization. Clear? So once this thing happens, the process is referred as nucleation, and once nucleation is induced, the ice crystal will form. Clear? And the plant will go under freeze stress or freezing state. Next comes tolerance. The first most, rather let's go for acclimatization. If you go for acclimatization, the first aspect comes in acclimatization is basic. As I said, here there are only two processes: either the plant resist it or avoid it. For avoidance, you can take very simple plants. For example, there are many species or herbs basically which grow in seas. If you are tolerant temperate conditions, they specifically grow in summer. They bear flower, they bear fruits, and they bear off. So they completely avoid the winters. Some plants, they are resistant in nature. So even if they are grown in cold temperate conditions, they will bear the you can say effect. One of the first condition which is adapted in the plants acclimatized for the cold weather is that the cholesterol content in the cell wall increases, or in rather terms I can represent the non-saturated fatty acids in cell wall increases. Once the non-saturated fatty acid is increased, the membrane will remain in a gel form for high or more time. The same thing reverse in case of heat stress. In case of heat stress, the tolerant plants or acclimatized plants will try or will tend to increase the number or concentration of saturated fatty acids in the cell wall. Thus, rather than being fluid in nature, the plant will remain in solid state, semi-solid state for higher amount of time. The same gets reversed in case of cold condition. The unsaturated fatty acids increases in case of plants. Clear? So that is acclimatization. The next is rather than storing water, the plants will tend to store acids more in concentration. Example I can give you is aloe vera. If you take an aloe vera, the uh, you can say liquid content in leaf is not exactly the water. It's rather you can say acids and organic acids. So that is first way of acclimatization. The next, if I go for tolerance, the tolerance is induced by AFP. The first aspect is AFP. Clear? That is, excuse me, give me a minute. So antifreeze protein, AFP. as in case of heat stress, again representing almost 60% defense mechanism in case of plants for heat stress, for cold stress will remain same. Iron, you can say transporters, then osmolytes, LEA proteins will remain as same as in cold stress as in heat stress. Uh, okay, I hope many. One question: which molecule is used in plants during cryopreservation to stop ice crystal formation in plants? Can anyone answer this question? Shall I repeat it again? Which molecule is used during cryopreservation to stop ice crystal formation during cryopreservation? इतनी शांति बर्दाश्त नहीं. Any answer? चलो. Anyhow, so basic AFP proteins, antifreeze proteins, osmolytes, proteins for membrane stabilization, and DIA proteins. As I said, many stress are interconnected in or interconnected with each other. The AFP protein, that is antifreeze proteins, which are induced in you can say plants in defense to the chilling stress, they act or they are you can say induced by ER genes. Correct? These ER genes are responsible for resistance to the genes which produce ER proteins, which causes defense against the pathogens. The same gene induces or you can say we can say traces for synthesis of AFP proteins. Once it is directed and AFP proteins are synthesized, these AFP proteins bind with you can say plasma membrane, or rather it binds with other membranes in the cells. Thus, it stops the effect of cold on membranes. In this way, it stops solid formation cell wall. The next is represent, we can say, induced factors for cold stress. The whole mechanism remains the same as that in case of heat stress, but rather than producing heat shock proteins, here antioxidants and COR genes are activated. These COR genes in you can say basically these COR genes are responsible for producing proteins which can resist the effect of cold stress in plants. Both the proteins or gene regulation in case of heat stress and the heat you can say gene regulation in cold stress is CAPK. Uh, it's based calcium based and excuse me. So they are you can see same transcriptional factors used by cis binding and say in, induction of many genes, which in turns will induce the synthesis of molecules, which will you can say stress protection. Okay, so basically that was about cold stress. Okay, so as we presented in most of them, heat stress, cold stress, they all lead to one aspect, that is you can say drought. The drought which is induced by both leads to the you can say activation signaling pathways, which in very simple terms is represented as drought related genes or drought related elements. Basically they are both cis binding elements. They induce the cis binding and ultimately leads to the transcriptional factors which releases enzymes, which enzymes releases proteins which sustain we can say the same kind of osmolytes are synthesized in case of solute stress, that is salinity. The same kind of osmolytes are being synthesized in case of drought stress. Synthesized osmolite, that is proline, in you can say any stress condition, be it heat stress, cold stress, acid stress or salt stress, water stress. The food produced convert into sugar alcohol most of the case. So in common it's sorbitol or mannitol. So in this way it stops the stress, or it increases the water potential, thus it helps in water retention in the dehydration caused by the heat caused by the salinity. Okay, water stress, the same embryogenic proteins will be synthesized and transporters will be synthesized, osmoregulators will be synthesized. So part of cold stress, part of heat stress, the only element is here heat shock proteins and COR genes will not be activated. So cold resistant genes and heat basic aspect comes is they synthesize different organs or they synthesize different material which helps them to prevent the impact or injury due to salt. A very simple terms which happens is most of the salt which is absorbed by the plants through roots are stopped at you can say endodermis due to the presence of Casparian thickening. So most of the straws salts in case of saline soil is stopped at Casparian strip endodermis. If it gets mixed in the cell and is absorbed by the xylem, then these salts will be stored in vacuole. Once its deposition in vacuole increases, it will produce a salt gland wherein all the salts through salt gland will be deposited on the leaf surface, and once the saturation period is achieved, the leaf will form. Leaf will form, fall. The rest of the mechanism remains same in as is in the other stress mechanisms. Okay. I have not incorporated any questions in my presentations for a simple reason no question is repeated in CSIR-NET. So to my suggestion, I'll request you all if you want to see the type of question what they have asked in last few exams, you can check the thing, but there will be no questions which is repeated. So I'll suggest you try to read the entire system as you can for the CSIR-NET preparation. I have presented here the weightage of representing the plant physiology is as it is in for different years. So starting from 2011 to 2019, the unit 6 has been marked for around, you can say, it ranges from 2 to 14, clear, in both part B and part C. If you take 2019, in case of December, 9 marks was from this unit, three in part B and 6 questions in part C. You can say in case of June, 3 questions from part B and 5 questions were from part C. So if totality, if I represent, it holds for a 50 concentration of marks. The questions or marks calculated here are having some common factors with other groups as well, as you can't change the topics or you can't completely separate the topics, so they may have one or two topics clubbed with it. In a way, I have considered them in my part. Anyhow, so that was the first suggestion for. If I say my topic representation, that is plant physiology, it includes photosynthesis, respiration, nitrogen metabolism, photobiology, movement of solutes, secondary metabolism, and stress physiology. Stress physiology, I had just covered the abiotic factors. For photosynthesis, you can say the questions are mildly easy, so you can just brush up your context. Same is in case of respiration. Most of the time, it comes in you can say part cell cycle, or rather respiration, different cycles in respiration. It comes from Krebs cycle. It comes from, you can say, glycolysis cycle. It comes from Kelvin cycle. They partly asks the enzymes or the intermediates synthesized during the process. For nitrogen metabolism, in last few exams, they have described or they have asked for the nod genes. So basically, nod A, B, C, D, E. There are 5 factors which are present in the nod genes, which translates different protein responsible for different aspect. In last few class papers I had seen, they have been asking, you can say, for nod genes, so you can brush about them. Nod genes, in case of plant hormones, they are mostly seen targeting jasmonic acids, racinosteroids. So again, with the same advent, they are targeting defense mechanism for the plant hormones. The next comes photobiology. this is the part where they have asked a very deep question. So if you are reading, you need to study this aspect in depth. Movement of solute, again you need to brush this thing up. Secondary metabolites synthesized, they are mostly targeting alkaloids. In alkaloids, they are you can say representing glycosides. So phenol, you can say you can brush up for the cardiac glycosides, rather sorry, not cardiac glycosides. You can brush up for the glycosides and terpenes rather terpenes and glycosides. One more thing in case of plant hormones is you can say Jasmonids. I represented it. So they are been asking Jasmonids. The last stress physiology. Stress physiology. They have again asked genes aspect of stress physiology or the molecular aspect of stress physiology. You can again recheck the or reread the. You can see the presentation as well as you can read the presentation. Okay. Now I open the channel to you all. If you want to ask any question, you can ask it now. Any questions?
Sir,
Ah,
uh, I just wanted to ask you one thing, that in this all these biotic and abiotic factors, like is there any possibility that due to some or the other problem or some other factor like chemical or anything, due to which the plant can get mutated and cannot be stress tolerant? Like for example, if we consider the abiotic factor for cold, the stress which is induced due to cold, but
hmm,
what if there are certain
hmm.
mutations in the plant key that cannot be tolerant or that cannot express the things which you explained during the cold? Are there cases like this happening in the plants?
Yeah, there are many cases. As in, if in natural we are not able to detect them for a reason, if they will be there or if such kind of mutation would have happened, then the plant is by default will be dead. So
Okay.
we won't get that plant. But in laboratory conditions, they have performed this kind of activities wherein the genes are you can say mutated, rather silenced, and once the genes
Okay.
are silenced, the plant make a defense or you can say kind of activity for defense or tolerance
So, sir,
for a very
ultimately
simple.
it will change in the regulation of the plant only, right? Due to this kind of mutation, like if it is not able
Yes,
to do the factors, I mean, if it is not able to incorporate the factors in a right way, then
definitely, definitely. And
okay,
what
yes,
you are
sir.
saying, we have studied that. And for that basically why we are representing the genes name or we are specifically indicating heat shock protein genes or genes responsible for
Yes, sir.
synthesis of heat shock proteins is because we have studied them by upregulating as well as downregulating them. So if
Yes,
they
sir.
are mutated or they are not functional, then heat shock proteins will not be synthesized.
Yes, sir. So,
So
sir,
yes,
the knockout and knock-in policies can be done to identify the genes also, which are responsible for the stress tolerance in different
yes,
factors.
yes, they
Okay,
can
sir.
be,
Yes, sir.
and
Thank
they
you.
are.
Yes, sir.
Welcome.
Thank you.
Okay, so any other questions anyone want to ask? Okay then. Uh, So with this, I end my session. I have uploaded my email ID. If you want anyone have any question regarding this, my email ID is rahulp@nuv.ac.in. You can bring on your questions and we'll definitely solve the questions for you. Okay. Uh, I'd like to thank all the Cheers PTM for organizing this activity. I'd like to thank Dr. Elizabeth Robin, and like to thank Dr. Darshi Bakshi for organizing this very good aspect for students to enrich their knowledge regarding Cheers PTM. Thank you, ma'am.
Okay, so I think Rahul, you are done with the session, right?
Yes, ma'am. Yes,
Okay.
ma'am.
Anyone has any doubts?
I had a
No,
question.
I think some of them have left. So, okay, so we will send the
Okay,
schedule
yes.
for tomorrow's session. We have two sessions tomorrow, so both the sessions details will be sent to you on email. And we thank Dr. Rahul for his session and also his expertise in the field. Okay, thank you, Rahul. And I think we'll end the meeting here and
Thank you,
we'll
ma'am.
see everyone again at 10
Sure, ma'am.
p.m. tomorrow. Okay, bye.